First of all, thank you very much, Patricia, for this very nice introduction. Thank you very much for all of you to be here. So as Patricia told you, I just want to say a few words before we start our real talk. As you might have noticed, Claudia didn't come because of COVID. And so to make the dynamics of our talk easier or better, we decided to make a short video. So just after my presentation, we will see the video and then we can discuss, okay? So first of all, I'd like to thank all the organizers of this very nice thematic trimester. So Luis Almeida, Vincent Cavez, Marie-Dominique, Benoit Partam, and Patricia Renaud-Bourret. And also the organizers of this very nice conference on mathematical neuroscience. So Suzanne Detelivsing, Olivier, Antonio, Patricia, Shinomoto. And also I would like to thank the IHS and also IHP for providing me this very nice environment to do mathematics and try to understand some questions that I'm very interested in. So, okay. So what is the context of my research? So as you know, often if you look at the some project in neuroscience, you have a sentence like neuroscience is often referred to as data-rich, but theory poor, right? So this means essentially that nowadays we have an excess of a massive amount of data, but still we observe many interesting phenomena on this data, but we usually they are poorly understood, right? So we need to do some theory or develop some statistical tools to make sense, to account for those observed phenomena. And this is essentially the content of my project. And what I want to do specifically is to try to answer two types of questions. The first one is what is written there to develop a new mathematical framework. We study the population coding for grid and place cells. So what does it mean? So in those two pictures, what you see here is the trajectory of a rat in a square environment in black, okay? And in the picture in both pictures, the red dots, they correspond to positions where a given neuron is more active, is spiking more, okay? And so in the picture on the right-hand side, you see that those positions are much more localized, and cells and neurons with this property are called place cells, okay? And the region is called place field. And on the picture on the left-hand side, you see that the structure of those prefed locations is much more richer, right? And in particular, they form a hexagonal grid, okay? And that's why they call those cells grid cells, okay? And one of the questions or the type of questions that we would like to answer here is, okay, first, we can think of as these two population of neurons as coordinate systems of the physical world in the brain of the animal, okay? And what we want to do here somehow is to say, okay, I take two positions, how close they can be, and in such a way that we still can distinguish between these two positions just by looking to one of these two populations, okay? And also, we would like to say if that the coding that we see here is optimal in which sense, okay? And so we want to formalize this in terms of hypothesis testing, which is very different from what people have been doing to study this type of cells. They have been doing using tools from information theory and all, and we want to say something different and try to compare with the results that we have so far. Okay. And the second type of questions is, okay, so usually, so the picture here is just to remind us that most of the times, or always I would say, when we record the activity, the spiking activity of a given animal in a given experiment, it turns out that activity is always, the spike activity corresponds to a very small part of the brain, right? So in this sense, the information that we have is always partial, okay? And the question is that we want to address here, okay, when we have it, we look at this data, what can we actually learn from them, okay? Especially because the information is partial. And it turns out that even, I think it is just everybody is aware of this fact. When we go to the literature, you see that many of the statistical methods are not tailored for partially observed networks. So there are a few exceptions, and probably there are others that are missing here, so I apologize. I just want to say a few of them. So for instance, we have this paper from Mathieu Lehalle and Daniel Takahashi, where they have a Markov random field. They want to estimate the transition probability. They know that they observe just the activity to a given small region of the whole space. But here it's difficult to apply this in neuroscience because they speak about IID models, which is an assumption which is difficult to justify when we look at the data, right? There are also there is also a paper from Liederman, Adams, and Pillen where they have this Bayesian approach for a GLM model, which is also they want to model partial observed networks. But it turns out there is no theory there, okay? And then there is a paper from Aline, Antonio, Eva, and myself when we have a spike network of neurons. And we wanted to estimate the graph of interactions. We still have the results, which we have some results for a partial observed case. But it turns out the method, the algorithm is not computationally efficient, okay? And then more recently, there is a paper from Liu, where he studies Hox processes, which have been used a lot in neuroscience the last year, sorry. And he wanted to, so he has a random network, and he wants to estimate the density of connectivity in this graph, in this model. And even in the partial observed case, and it turns out, I mean, the downsides of this paper is that the intensity is linear, and if you know a bit about Hox processes, you realize that in this case you can only have excitatory connections, and so this is a bit not well suitable for data, let's say, data analysis. And the last work that I would like to mention is a paper with Patricia and myself, where we have this large network of neurons, even an infinite network of neurons, and we wanted to estimate the transition probability, right? I mean, the probability of observing a spike given the past. We want to estimate this function. And we have some results to approximate this transition probability by using some given dictionary. The theory here is nice, but the problem is, as you might know, when we have these lasso results, we always rely on these restricted eigenvalue properties, which you need to check for each dictionary that you are using, and this may be difficult depending on the dictionary that you want, okay? And I'm sure that, so this is more or less related to statistical analysis of neuronal data. And I'm sure that we'll hear much more about this in the conference by Shinamoto, Eden, and Cass. Okay? So just to conclude, I would like to mention that I've been working a bit also from, let's say, in the modeling of large and spatially extended networks of neurons. And one usual approach to do this is by means of mean field analysis. So you have a model, you give a description of how your neurons interact at the macroscopic level, and then you try to derive a macroscopic limit or equation to which will depend, of course, on how you impose the rules at the microscopic level, okay? And so I have worked with Aline, Andres, in a GL model. I'm sure that we heard about this model during the week. And more recently, I have worked with Julien, Eva, and Aline also when we have this a model for Hox processes with spatial where the interactions taking into account also the spatial positions of the neurons. And we show that this process, so if I have a Hox process at a microscopic level, then at the macroscopic level, we ended up with what's called a neural field equation. And on Friday, I think it's Fridays, or at least the last 2 days are dedicated to objects like this, okay? And finally, I would like to mention that we also have studied the fluctuations of these spatially structured Hox processes around its limit, which is a neural field equation, and then we make sense of what's called a stochastic neural field equation. And we would like, in this context, we'd like to understand how we can use this result, for instance, to estimate the connections of your network. Okay. And then, as I said, we will heard much more about GL neural models during this week. So we'll have the talks by Fumier, Rush, Lechekba, Ipuzar, and also Morgan. And we'll heard much more about Hox processes and neural field equations in the Phi, Stanat, and Phase talks. Okay. So now, now I'd like to introduce our my joint talk with Claudia. Okay. So before we start, I just want to mention that this is a joint work with Noslin, Aline, Ricardo Freiman, Antonio Galvez, and of course, Claudia Vargas. And I hope you enjoy. And after that, I will be happy to discuss together with Claudia. Okay. So.
I would like to thank Patricia Bourret and the organizers of the workshop on mathematical modeling and statistical analysis in neuroscience for inviting Guilherme and myself to be here with you today at the Institut Henri Poincaré. I also wish to acknowledge the support of our funding agencies and that of FAPESP to the Center of Research, Innovation and Diffusion in Neuromathematics, Neuromat. The title of our talk is Retrieving the Structure of Probabilistic Sequences of Auditory Stimuli from EEG. This talk is part of the activities developed within the research project termed the statistician brain. According to this conjecture, acting as a statistician, the brain makes predictions about future events in order to interact efficiently in the world. Predicting means anticipating outcomes. The idea that the brain makes predictions so as to anticipate outcomes is by no means new. It goes back to the pioneer work of Hermann von Helmholtz, a 19th century multi-scientist who introduced the notion of unconscious inference. Helmholtz conjectured that inferring about future events should be a crucial mechanism for learning and memory formation. Since then, it has been widely proposed that the brain assigns probabilistic models to samples of stimuli, also called the Helmholtz heritage in the contemporary neuroscience domain. Our goal here is to tackle the following challenge. Does the brain assign probabilistic models to sequences of stimuli so as to act in the world. Take, for instance, a sports person like Fabiana Müller, our Brazilian world champion in pole vault. As stated by Helmholtz, she improved her fantastic skills by training and adjusting her models of the reality. But can we transform the sequence of actions performed by this incredibly skilled person into a mathematically treatable object? And how to approach this question experimentally? A typical solution to this problem in neuroscience is to employ, for instance, simplified sequences of visual stimuli, such as the one depicting a human walk. As shown in this picture, it is possible to put a marker in selected points of the body and then extract only the relevant information that relates to the body movement. We then present to participants videos depicting these point slides while we record their brain signals employing an EEG machine. These EEG signals can be treated so that each recording electrode is considered as a node in a graph of interactions, depicted here as red points, and the vertices correspond to interactions between nodes. We then analyze statistically the graph metrics per node, such as distance, degree, etc., and we get very interesting results about how each node behaves in regard to its relatives during the point-like display visualization. Note that there is an ever-changing activity through time across the graphs per participant, as depicted here for 2 repetitions of the same video. However, this approach precludes to retrieve from the recorded brain signals a signature of the employed visual stimuli. In other words, there is no way to establish a formal relationship between the sequence of point-like displays and the graph of interactions. So, at this point, with the Neuromath team, we had some open questions. How to extract from brain signals the very structure of a sequence of events? How to establish a formal relationship between this sequence of events and the recorded signals? We started to think on a new experimental protocol in which we would provide to the brain a model containing information about the structure of a sequence of events while we recorded the EEG signals. We wanted a model that could be described formally so as to retrieve this model from the EEG signal. The chosen model was based on the work of Jorma Rissanen, who proposed in 1983 a class of stochastic models capable of compressing any sequence of symbols generated by a source. Rissanen introduced a new class of probabilistic models, namely the class of stochastic chains with memory of variable length, also called context-free models. The mathematical formalism and the experimental approach employed in this work is described in this recent publication and will soon be explored in detail by Guilherme. Shortly, volunteers were exposed to sequences of auditory stimuli where they had their EEG signals recorded. Stimuli consisted in strong, weak, and missing hand claps. We employed 2 different context trees that were called ternary and quaternary. In the ternary sequence, a strong beat was followed by 2 weak beats, and in the quaternary tree, besides, there was a constitutive silent unit separating the 2 weak beats. In both cases, eventually, the weak beat could be replaced by a silent unit, introducing a variability in the sequence of events. Following Rissanen's proposal, these sequences can be represented by context trees. Given the present symbol, the context tree represents all the possible past sequences with respect to that present unit. The transition probability associated to each context is used to choose the type of auditory stimulus appearing after that context in the sequence of stimuli. When we look at the EEG signal recorded during an experiment, there seems to be not much information across electrodes, as shown here in the right. In the neurophysiology domain, a trick is to repeat the same stimulus, say 1,000 times, and average the corresponding EEG response segments. By doing this, we get an event-related potential. The auditory event-related potential has certain regular temporal features allowing its identification. If a sequence of identical auditory stimuli is followed by a deviant sound or even by the auditory stimulus absence, there is a specific modulation in the EEG signal that is called mismatch negativity response. This marker of novelty, discovered in 1978 by Naatanen and colleagues, has been reported since then by several researchers in the field. Here, the deviant response is depicted in red, as opposed to the response to a standard auditory stimulus depicted in blue. 0 on the X-line corresponds to the stimulus onset. The red spot in the brain image in the right shows the location from which the mismatch response was recorded in this participant's brain. In this case, the left auditory cortex. As you will see, the mismatch negativity effect helped somehow to guide our analysis.
Hello everyone, my name is Guilherme, and in the next few slides, I will show you how we model the auditory stimuli we consider through context tree models and how we characterize context tree models. To this end, let me start with this simple example. So consider the following deterministic sequence 2, 1, 1, 2, 1, 1, etc., where the symbol 2 corresponds to a strong bit and the symbol 1 corresponds to a weak bit. Then replace each symbol 1 by a symbol 0, which from now on corresponds to a silent unit, and we do this independently with probability 0.2, producing, for instance, a random sequence that you see here on the screen. So in this case, it's 2, 1, 1, 2, 0, 1, 2, 1, 1, etc. This random sequence is an example of what we call in probability theory stochastic chain. And the stochastic chain we produce in this way is going to be denoted by X in boldface. And this is a shorthand notation for the whole sequence, which is X0, X1, X2, etc. And in this example, X0 is the symbol 2, X1 is the symbol 1, X2 is the symbol 1 as well, and so on. And by the structure of this auditory stimulus, we call it ternary. Now, let us observe that there is an alternative way of generating the ternary chain X. The chain X can be generated step by step, and this can be done as follows. Suppose we want to generate Xn given the past Xn minus 1, Xn minus 2, etc. What we do is first we look at last symbol Xn minus 1. If Xn minus 1 is equal to 2, then Xn, by the structure of the chain X, is going to be 1 with probability 0.8 or 0 with probability 0.2. Now, if Xn is either 1 or 0, then we need to go back one more step in the past. So let's say that at time n minus 2, we have a 2, that is Xn minus 2 is equal to 2. Then, by the way we constructed the chain X, we know that Xn is going to be 1
with probability 0.8 or 0 with probability 0.2. Why? So remember, we want to simulate xn, so at time n we have a question mark. We want to simulate the symbol xn. And in this case, we know that we have observed at time n minus 1 either 1 or a 0, and at time n minus 2 we have observed the symbol 2. So by the first way we constructed the chain X, we know that at time n we will observe a 1 if we didn't erase the deterministic 1, and this happens with probability 0.8, or we will observe a 0 if we replaced the deterministic 1 by a 0, and this happens with probability 0.2. The other possibilities for xn minus 2 is either the symbol 1 or the symbol 0, and by the structure of the chain X, we know that in this case, xn will be the 2, so that's why I have here xn equals 2 with probability 1 in this case. And we have a small drawing here on the right-hand side, just to explain this again. So we are at time n, we have a question mark here, we want to simulate xn, and at time n minus 1 have observed either 1 or 0, and the same thing for at time n minus 2. So we have observed either 1 or 0. And by the way we constructed the chain X, we know that at every 3 symbols we observe a symbol 2. So in this case, we must observe a symbol 2. The algorithm generating X is characterized by 2 elements. The first one is a partition of the set of all possible sequences of path units that we denoted tau, and this set tau is the set of the strings 0, 0, 1, 0, 2, 0, 0, 1, 1, 1, 2, 1, and 2. In the partition tau, for example, the string 0, 0 represents the set of all past symbols ending by the ordered pair 0, 0. And to give another example, the string 1, 1 represents the set of all past symbols ending by the ordered pair 1, 1. Following reasoning, the elements of tau will be called from now on contexts. Moreover, this set tau can be represented by a labeled and rooted tree, as shown in figure 1. As you can see, in this tree, the time goes downward. Each level of the tree represented a different time instance. Moreover, the leaves of this tree are the contexts of tau, and this tree representation gives rise to the name context tree. So that's why we call tau an example of a context tree. In the algorithm generating X, for each sequence of past symbols, the algorithm first identifies the corresponding context, W in tau, and once the context W is identified, then the algorithm chooses a new symbol A in the set 0, 1, 2, using a transition probability PA given W. In other terms, each context W in tau defines a probability measure on the set 0, 1, 2. Thus, the family of transition probabilities PAW with A in 0, 1, 2, W in tau is the second element characterizing the algorithm generating X. The family of transition probabilities associated with tau are shown in the figure 2 on the right-hand side of the screen. And to give you an example, the transition probability indexed by the context 2 is given in the first row of the table appearing in the figure 2. As you can see, P0 given 2, that is the probability of observing a 0 after observing a 2, is going to be 0.2, and P1 given 2, that is the probability of observing 1 given that we had observed a 2, is 0.8, whereas P of 2 given 2 is going to be 0. That is, the probability of observing a 2 given a 2 is going to be 0. Okay? So these are the 2 elements characterizing the algorithm generating the chain X. The ternary chain X is an example of what we call a context tree model. An important feature about context tree models is that the dependence from the past has not a fixed length. Instead, it changes at each step as a function of the past itself. This explains why this class of models are also known as stochastic chains with memory of variable length and variable length Markov chains. The class of context tree models have been introduced by Henri in 1983 as a universal system of data compression. And very briefly, a context tree model is a stochastic chain characterized by a pair tau p, where tau now is the context tree describing the dependence from the past of the model, and P is the associated family of transition probabilities indexed by the context tree tau of the model. If you want to know more about context tree models and their applications, here we have a few important references. The first is from Henri with applications to information theory, and then the second one is from Bierman and Weiner with applications to DNA data. And then the third is from Galvis and et al. With applications to linguistics. And the last one is from Belloni in Buzzero, also with applications to linguistics. We also used another context tree model to generate auditory stimuli in our study called quaternary. The quaternary auditory stimuli is obtained by considering the deterministic sequence 2101, 2101, 2, etc. And then very similarly to what we have done to the ternary auditory stimulus, we replace in a IID way each symbol 1 by a symbol 0 with probability 0.2. By studying the algorithm used to generate step-by-step the quaternary auditory stimulus, one can check that the corresponding context tree is the one that you see here on the left on the screen, and the family and the associated family of transition probabilities are given in the table that you can see here on the right-hand side of the screen. Recall that in the experimental protocol Claudia described before, we have volunteers listening to a sequence of auditory stimuli while EEG signals are recorded from their scalp. We have seen so far how we model the sequence of stimuli as context tree models, and now I will explain how we model the EEG signals associated to a given sequence of stochastic stimuli. To this end, let Yn be the chunk of EEG signal recorded while a volunteer is exposed to the auditory stimulus Xn. For instance, in the image here on the screen, we have an EEG signal of a given electrode, and if you look at the green rectangle, we have that Xn is equal to 2 for a given suitable n. In this case, Yn is the chunk of EEG inside this green rectangle. The time domain of each EEG chunk is 450 milliseconds, from -50 to 400 milliseconds relative to the onset of the auditory stimulus. The model assumption is the following. The law of Yn depends only on the context W associated to the past Xn, Xn-1, etc. According to this assumption, for instance, whenever the context associated to the past Xn, Xn-1, etc. is 2, that is, Xn is equal to 2, the law of the EEG chunk Yn is given by a suitable distribution indexed by the context 2. Now, if the context associated to Xn, Xn-1, etc. is, let's say, to 1, then Yn is chosen according to some distribution, which is going to be indexed by the context to 1, and so on. The EEG signal is then the stochastic process Y obtained by concatenating the EEG chunks Y0, Y1, Y2, etc., constructed in the way we just described. From the statistical point of view, one of the main contributions of our work is to present a statistical procedure to estimate a context tree from example x0, y0, etc., xn, yn, where x0, x1, xn is a sample of auditory stimuli modeled by a context tree, and y0, etc., yn are the associated EEG chunks. And this can be done as follows. We fix first an integer k larger or equal than 1, and we do the following. 1. For each symbol A in 0, 1, 2 and string U of size k of symbols in 0, 1, 2, we define the subsample of all EEG chunks ym such that xm minus k is equal to A, xm minus k plus 1 is equal to u1, etc., xm is equal to k. So to give an example, in purple here, we have examples of k equals 2, u is the string 1, 2, and A is the symbol 1. And the rectangles, and the green rectangles corresponds to examples of k equals 2, u equals 1, 2, and A equals 0. In the second step, we do the following. For any pair A and B, we test the null hypothesis that the law of the EEG chunks YAU and YBU collected at step 1, not 2, so this is a mistake, are equal. So here there are two possibilities. The first one is if the null hypothesis is not rejected for any pair of final symbols A and B, we then conclude that the occurrence of A and B before the string U do not affect the law of the EEG chunks. And in this case, we repeat the procedure with the shorter string U, which is U2, etc., UK. And the second possibility is if the null hypothesis is rejected for at least one pair of final symbols A and B, we conclude that the law of the EEG chunks depend on the entire string AU, and we stop the pruning procedure. In step 3, we keep pruning the sequence U1, UK until the null hypothesis is rejected for the first time. And in step 4, we call tau hat n the tree constituted by the strings which remained after the pruning procedure. An important step in the statistical procedure I just described consists in testing the equality of distributions for two samples of EEG chunks or segments. To this end, we used the projective method proposed by Cuestas Albertos et al. In 2006. In a few words, the projective method works as follows. First, we choose at random a direction, for example, a Burnham motion or a Burnham bridge, and project each function in the samples in this direction. And this produces two new samples of real numbers. And in the step 2, we test whether the samples of the projected real numbers have the same law or not. And we can use, for instance, here at this point, the Kolmogorov-Smirnov test. Under suitable conditions, it has been shown in Cuestas Albertos et al. That for almost all directions, if the test does not reject the null hypothesis that the projected samples have the same law, then the original samples have the same law as well. The fact that the result holds for almost all directions allows us to repeat step 1 and 2 above for several directions. In this case, we decide not to reject the assumption that the two samples of EEG have the same distribution if the number of non-rejections of the projected samples over the different directions is sufficiently large. In our paper, we used in each test 5,000 directions, and we only rejected the null hypothesis that the distributions are the same if for more than 276 rejections are obtained over the different directions, and we refer to our second paper to the details justifying this choice. After applying the statistical procedure described above, per electrode and per volunteer, we obtained a tree denoted by tau hat n e v. So this is the context tree estimated in the electrode E of volunteer V. And by doing this, we ended up on each electrode E with a set of context trees that we denote tau En, as you can see here in step 1. We then summarize this set of trees in a unique tree called mode context tree, which is defined as follows. We compute the number of times each string W appears as a context in the context trees belonging to the set TEN. The context in the mode context tree, marked in red here in the image on the right-hand side, are the nodes that satisfy two properties. The first one is no node in the subtree induced by W has greater value than it. And two, no node in the path from W to the root satisfies the property 1. Let me give you an example. In the image displayed on the bottom right of the screen, the string 00 belongs to the mode context tree because the nodes 00, 100, and 200, which are the nodes in the subtree induced by the string 00, they appear 4 times each, whereas the string 00 itself is estimated or appears as a context for 11 volunteers, as indicated in the number in red. And so this is property 1. Moreover, the nodes in the path from 00 to the root are estimated as a context for no more than 4 volunteers, as indicated in the picture. So the string 0 was estimated as a context for 4 context trees, and the root was not estimated as a context for none of the estimated context trees for that electrode. One can check that in this example, the nodes in red are indeed the context of the mode context tree, as we just did for the string 00. Very similarly, one can check that in this example, the nodes appearing in red are indeed the contexts of the mode context tree. And we can also see in this example that the mode context tree matches perfectly the context tree of the ternary chain, which was the true stimulus in this case.
So here I show the results for the quaternary context tree. This picture shows a sample of results obtained for a given electrode per participant for the quaternary context tree. We had 20 participants in this experiment. You can appreciate that there is some variability across participants. Similar results can be appreciated across participants for the ternary tree. Some variability can be identified across participants. A challenge of this work was how to average these trees across participants per electrode. As detailed by Guilherme, this was solved by estimating a mode context tree per electrode. This is a summary of our main results, published recently in scientific reports. Employing the mode approach, we were able to reconstruct a context tree per electrode across participants for the quaternary tree in the left and the ternary tree in the right, here represented as top views of the skull. The color code consists in a distance measure from the original context tree, where green corresponds to maximal similarity and pink maximal dissimilarity from the source context tree. As you can see, signals recorded from frontal and left temporal electrodes, labeled in green, were those who were closest to the original tree. These results are consistent with fMRI studies showing that the prefrontal cortex is involved in learning context-based statistics. So to conclude, we established the formal relationship between the structured sequences of random stimuli and the stochastic process associated to the brain processing of those stimuli. This was done by employing the new functional data statistical selection procedure just described by Guilherme. The obtained results support the conjecture that the brain effectively identifies the context tree characterizing the source. Guilherme and I wish to acknowledge the colleagues that participated in this work. Thank you for your attention.
Thank you a lot, Claudia and Guilherme. I think there is time for questions now. Is there any question in the room? I have one. At the beginning of Claudia's talk, you were speaking about connectivity between the electrodes. And then your analysis for the moment is just analysis electrode by electrode. Do you have any idea how to take into account this connectivity between electrodes or brain areas inside the analysis?
Yeah, that's a great question. I don't know if I'm... okay. That's a great question. For the moment, I at least didn't think about how can we try to account for this, for what you mentioned. But maybe, I don't know, Claudia may have... may want to say a few words about that.
Yes, thank you very much, Patricia. Excellent question. We have been considering this possibility, but we didn't work yet on this direction. But I think it's an excellent suggestion. We can move forward on trying this approach. Thank you.
Especially the trees you showed at the end, they seem really different depending on the electrode. And if I understood correctly, the green ones are the trees that are the closest to the tree of the signal, right?
Mm-hmm. Right, exactly.
Exactly. Do you have any idea, for instance, why the ternary and the quaternary rhythm do not even involve exactly the same electrodes? What would be the cause of that?
Guilherme wants to discuss on that. Patricia, that's probably a mixture of the procedure that we have employed. But what is similar between them is that we do have the full trees reconstructed in the prefrontal electrodes, not all of them for the ternary, as you noticed, but the fact that we have a constitutive silent unit in the quaternary tree may have helped us in identifying more properly the original tree. So this is something we can discuss more.
Okay, thanks a lot. Another question.
Yeah, thank you very much for the presentation. Very interesting. Indeed, you have isolated a very interesting effect. My question was, how would you, do you intend to generalize to more complex types of stimuli, and how does it fly for more complex things?
Yes, very good question. Yes, we are now starting to build different types of context trees. For instance, the trees that we have employed have a regularity, a regular base, a structured base. So it will be interesting to see what happens if you break down this regularity, this periodic, say, structure of the tree, and see how it goes for participants to learn this different structure. So we are now interested in going more on depth on the kind of stimuli that we have just built, Olivier. So this is our proposal for now.
Yeah, I just wanted to mention also that I know that there is, in Neuroma, there is an ongoing project where they have a similar protocol where they have a person playing a game. And then the game is essentially, Claudia can correct me, of course. I may say something wrong here. And the idea is that, so you are the goalkeeper, and there is you are the goalkeeper. So here, you are the goal, and then there is the kicker here. And the strategy of the kicker is given by a context tree. And then the person is playing. And then, of course, now the data is not anymore the EEG, but they are measuring the reaction time of the goalkeeper. And the idea, so again, you have either the left, right, or center. So that's the three possibilities. But the protocol is essentially the same, and so the idea is that try to by looking into the answers and the reaction time, if we could try to retrieve also the tree in the same spirit as we did here. So just to mention that there is already another ongoing project where they have different type of stimuli applied. Okay, so probably we should have a link here, but if you type goalkeeper game Neuroma, you'll find much more about this project. Okay.
Yes, excellent, Guilherme. Thank you for bringing up this protocol that we are working also, and we wish to couple with EEG. This is the next step. Also, couple the goalkeeper game with the EEG, so that we can start looking at these prediction markers also associated to making a decision to go left, right, or center, and learning this sequence of events. Yes, thank you.
Other questions? Yes.
Hi, thank you for your talk. I'm neurobiologist, so I didn't understand all the mathematical part, but so I was wondering, when you were talking about this task where is motor task, okay? But so you were also mentioning about direction and decision making. Do you think that you will have a signature on the EEG, like a mismatch, like you were explaining, the one that you were explaining that is very, very clear to detect? But for the decision making, I don't think that you will have only one signature visible on EEG. And so what is your strategy in this case to solve this problem? Because it's all our problem in biology. So we have many cognitive part, and so it's not only just action and movement.
Yes, very interesting question. Thank you. So the idea would be to decompose, to choose the windows of over the EEG that corresponds to the moment the person chooses which will be the next step. And by choosing, he has to press a button. So we have this period just before the response selection. And then the game is structured so as he has a feedback of if he has given the good response or the bad response just afterwards. So we can just cut the EEG signals in periods and trying to find the specific signature that corresponds to the response, the prediction says associated to the response that the person gives, and then the feedback he has afterwards. I don't know if I made myself very clear, but if I answered your question, you want to comment on that?
I really understand, yes. That means sequence the different process step by step and see if you have a signature for each of them. Is it?
Yes, exactly.
Right.
Yes,
Thank
yes. Thank you.
Thank you. Other questions?
Okay, thank you very much for this nice talk. I have a question rather related to this decision-making problem and a little bit similar to the previous question, related to the previous question. You mentioned that in the other project, which covers studies more or less a similar approach, but applied to this goal-keeper problem, that the indicator that was considered was rather the reaction time. But the relation...
I'm sorry, go on. Sorry. Yeah.
Okay, but the relation between the input, let's say, in that problem and the output, in my understanding, is reflected rather on the decision, the final decision which is made, the performance result.
Mm-hmm.
So, and also maybe even this performance result is not sufficient, or only reaction time is not sufficient, and maybe other indicators are needed to be checked. And this was my question: how using only the reaction time, you think, shows or reflects the relation between the input and the output of the whole procedure of decision making? It's also quite high-level procedure process in comparison to...
Yeah, of course. I mean, this is of course. I will rely much more on Claudia's answer because I'm not really involved in the project. I just wanted to mention that there were another ongoing projects with close type of proposal that we were just describing. So I don't know, Claudia, if you want to answer the question.
Yes.
I don't know.
So we are looking at the performance of the participants. So the error rate, and this information gives us a measure of how learning evolves through time, of course, how the person is learning that context-free model, right? And we also are measuring the reaction time, which surprisingly gives us interesting information on how the person takes into consideration the errors that he made in the past. So this was just published as a doctorate thesis in our Neuromat, work done by Paulo Passos. And the next step now is to use information coming from the EEG while the person is doing the task, so that to try to disentangle these several say contributions of the moment the person makes the decision, makes the choice, and also how he reacts to the error that he made. So this is an ongoing project. Thank you.
Okay, thank you.
Okay, thank you. I think it's time we switch, but it was very interesting, and thank you both of you for doing the opening of this conference. Yes, we clap. That's the time.